Good morning to all of you. Today I am going to talk about epistasis. It is one of the important topic of CSIR NET for life science because in alternate years usually questions come from such particular topic. So what is epistasis? I will discuss. Apart from what is epistasis, definitely I will try to show that what kind of epistasis actually exist and how it is actually simply you can say the modification of Mendelian ratio, okay, especially 9:3:3:1. So what is called epistasis? Epistasis may be defined as is one of the typical genetic phenomenon where the function or certain characteristics of certain gene has been masked or suppressed by another gene, and definitely epistasis is actually definitely occurred as for interallelic interaction. Okay, so you can say an interaction between a pair of loci in which the phenotype or phenotypic effect of one locus depends on the genotype at the second locus. So definitely you can say genes whose phenotype are expressed definitely epistatic, and gene whose phenotype actually altered or suppressed, it is called hypostatic. Now there are so many differences between dominance and epistasis. What are the difference? In case of dominance, actually you can say it is intraallelic interaction, but epistasis is actually interallelic interaction. On the other hand, in case of dominance, one allele hides the effect of other allele at the same gene pair. What about epistasis? One gene hides the effect of other gene at different gene loci. As you all know, a gene is a chemical determinant. Gene products interact with the environment and factors such as temperature, light, hormones, and enzyme. If there is any problem or mutation in the intermediates, it can definitely lead to another phenotypic, and it is called definitely disturb the Mendelian ratio. Next, here is a classical example actually of regarding epistasis, the eye pigmentation in case of Drosophila. There are mainly two different kinds of genes that actually exist. One is called vermilion, mainly quoted by small v. Another is cinnabar, small cn. Now the flies that are mutant for cn lack xanthomatin, it's a kind of pigment, and they have bright red eyes because of presence of drosophila. Though in some case actually mutant small v, mainly vermilion, flies also lack xanthomatin, but for a different reason. Now in these flies the pathway is blocked because there is no functional capital V in the vermilion enzyme. Now I will discuss there are different kinds of epistasis, or you can say epistatic interaction exist. Number one, dominant epistasis. The ratio suggests 12:3:1 because, as I have already told you, the epistasis is a kind of modification of typical Mendelian ratio 9:3:3:1. Now in case of recessive epistasis, it is 9:3:4. It is also called supplementary interaction. Number 3, duplicate recessive genes. The ratio suggests 9:7. It is called complementary genes or complementary interaction. Number 4, duplicate dominant genes. The ratio suggests 15:1. And duplicate genes mainly with cumulative effect, it is suggests 9:6:1. And lastly, dominant recessive interaction. The ratio suggests 13:3. Next, I will discuss one by one with classical example for better understanding. Dominant epistasis. The ratio suggests 12:3:1. Now, dominant allele, for example, you can consider capital A, of one gene hides the effect of alleles of another gene, for example capital B, and express itself phenotypically. The capital B allele definitely is hypostatic in nature because the functions of capital B has been suppressed by capital A gene, which is epistatic in nature. So thus the genotype, you can say capital B, capital A, and capital B, or you can say capital A, capital B, small b, and definitely on the other hand capital A, small b, or capital A, small b, produce the same phenotype. Now genotype capital A, small a, and capital B, and on the other hand small a, capital B, small b, and small a, b, produce two additional phenotype. Now this type of dominant epistatis definitely modifies the classical ratio of 9:3:3:1 into 12:3:1. It's a classical example of dominant epistatis studied in summer squash, because there are two types of color usually found. Definitely W suggests white is dominant over colored squash. On the other hand, yellow, capital Y, is dominant over green squash. Okay, definitely pure breeding white fruited variety is crossed with the double recessive green variety. F1 hybrids are all white. Now when the hybrids are self-white, yellow and green fruit plants arise in the ratio of 12:3:1. Now you can represent it with the help of, you can say, Punnett square. Recessive epistatis, the ratio suggests 9:3:4. It is called supplementary interaction on the other hand, because recessive alleles, for example small a of one gene locus, hides the effect of another gene locus and express itself phenotypically. So the alleles of capital B locus express themselves only when epistatic locus has dominant allele. Try to understand the basic difference. For example, capital A, and capital A, small a. So ratio will be modified into 9:3:4. There are so many examples. One of the classical example is, in case of horse, the brown coat color is dominant over tan. Also apart from this, there are so many examples out there. How we can represent next recessive genes. What about duplicate recessive genes? It is also called complementary genes. It's called 9:7, because both the genes loci have homozygous recessive alleles and both of them produce identical phenotype. Both dominant alleles are necessary to produce a different phenotype. For example, capital A, capital B, capital A, small capital B, and also other hand capital A, small a, capital B, small b, in all these combination. So both the dominant allele capital A and capital B are present and they will produce a different phenotype. So whereas you can say small a, capital B, or small b, capital A, in which the other dominant allele is absent, produce the normal phenotype. Complementary gene interaction or duplicate recessive gene. There are so many examples as usual. One of the classical examples is on sweet pea, because scientists Batterson and Punnett observed that when two white flowered varieties of sweet pea, the scientific name is Lacerus odoratus, were crossed, F1 progeny had colored flowers. When F1 was selfed, the F2 ratio showed the presence of both colored and white flowered varieties in the ratio of 9:7. Remember this? This is how we present it. Okay, and also as for remarks, in this case dominant alleles on both locus are required. Hence, whenever capital A and capital B both are present, they result into purple effect, masking the white. This is because capital A and capital B alleles modify the colorless precursor by showing their effects. What about duplicate dominant genes? The ratio suggests 15:1, and also such duplicate genes are called pseudoalleles. Remember the term pseudoallele. So dominant allele of both the genes produce the same phenotypic effect, giving the ratio called 15:1. At least one of the dominant allele is necessary for the phenotypic effect. Now, in the absence of all the dominant genes, only in case of definitely small a, small b, the recessive phenotype will be expressed. So, those duplicate genes are also called pseudo-alleles. Remember the term pseudo-alleles. For example, there are so many examples. One of the classical example actually of seed capsule of Shepherd parts genus Capsella occur in two different shapes. Okay. Next, duplicate genes with cumulative effect. The ratio suggests 9:6:1. Both the dominant non-allelic alleles, when present together, give a new phenotype, but when allowed to express independently, they give their own phenotypic expression separately. That's why it is called cumulative effect. Duplicate genes with cumulative effect একসাথে কাজ করে. Okay. As for example, you can say in case of capital S and small s are allelic genes. So capital A is giving sandy color usually and small s giving white color. Our non-allelic genes, capital R also gives sandy color, same as S, but the basic difference lies when both the dominant genes interact together, they give red color. And definitely you can say non-allelic genes doesn't interact with small s. Next, what about dominant recessive interaction? The ratio suggests 13:3, quite significant, because dominant recessive interaction 13:3. The dominant allele capital A either in homozygous or heterozygous condition of one allele gene, and the homozygous recessive allele capital small b of other gene produce the same phenotype. In F2 generation, progenies having capital A or small b will not allow capital C gene to be expressed. That's the basic difference. That's why it is called dominant recessive interaction. As for example, there are so many examples out there. Okay, in hornfowl, the white color of feather is formed by certain genes, mainly for example, capital C. 2, so due to the presence of epistatic gene 1. And lastly, as for summary, as for beta consideration, as for the remembering, you can say it's a classical chart by which you can remember all those ratio, because it is so important for NET examination. So at first, as for additive effect, because it is typical Mendelian ratio 9:3:3:1, and then comes to the complementary gene interaction where at least one dominant allele from each of the two genes needed for phenotype. So the ratio suggests 9:7. Then as for recessive epistatis, homozygous recessive genotype at one locus allele must or hide or suppress the expression at second locus. The ratio suggests 9:3:4. And then dominant epistatis, the ratio is 12:3:1, where the basic inheritance pattern suggests dominant allele at one locus must or hide expression at second locus. And lastly, what about duplicate genes? One dominant allele from either of two genes will be needed for the phenotype. The ratio suggests 15:1. It is all about the typical different modifications of Mendelian ratio. So it is called different kinds of epistatic interaction or epistatis. Thank you. And definitely I can tell you one thing, that particular topic is so important, as I've already told you in the beginning, and definitely will get questions from the particular topic called epistatis. Thank you so much.